Hi everyone, today we're going to be looking at the second lesson or the second section of this chapter, and that is DNA technology. Now, some vocabulary that we should know before is DNA, and we took this in term 1, and we know that DNA is the genetic material of all organisms, and it is composed of two complementary chains of nucleotides that wound into a helix. So before we talk about genetic engineering and what genetic engineering is, let's have a little look on the history as to why we do genetic engineering. So by 1970, researchers had discovered the structure of DNA, and we took that last term. We talked about the structure of DNA, and we talked about the central dogma or the DNA flow, and we said that is that DNA goes to RNA and from RNA to protein. Scientists knew that, but they didn't know about the function of individual genes. So an example would be if your friend told you that the high school team scored something in a football game, they scored high in a football game, but they didn't tell you how each player contributed or what each player did to get that win. So that's what happened with scientists. They knew that DNA goes to RNA and RNA to protein, but they didn't know what each gene does. And scientists were curious about that, right? They want to know what these genes are doing, what each gene does. And that's what gave us genetic engineering. So when a scientist started using genetic engineering, which is a technology that involves manipulating DNA, and when I say manipulating DNA, I mean cutting, pasting, cropping DNA. Okay, so we're cutting pieces, we're adding pieces, stuff like that. So we manipulate the DNA of one organism to insert exogenous DNA. And what does that mean? That means the DNA of another organism. Now, when they tell you exo, that means it's outside. So it's not the DNA of this organism. It is a DNA of another organism being inserted into this organism. So if I had, for example, a dog and a cat, and I was doing genetic engineering, what I would do is I'd take some strands of DNA from the cat, and these would be my exogenous DNA, right? Because they're not from the dog, right? I want to do the genetic engineering on the dog. So I'll take a couple genes from the cat, let's say, and then I'll insert it into the dog. So the cat's genes are exogenous. Okay, so again, let's look at what genetic engineering is. Genetic engineering is the technology that involves manipulating the DNA of one organism in order to insert the DNA of another organism. And that DNA that we're inserting is called exogenous DNA. All right. So we know what genetic engineering is, but what are genetically engineered organisms used for? Before I give you any examples, let's talk about what are they used for? Why do I need genetically engineered organisms? Well, these organisms can help in and I'll show you where to highlight on the book here. They can help with the study of the expression of a particular gene. So we can look at the studying of a expression of a particular gene. And when I say expression of a particular gene, I'm basically talking about how a gene works, or how we express that gene, that particular gene. What is the function of that gene, right? The second thing is to investigate cellular processes. And when I say cellular processes, I mean anything that's carried out at the cellular level. So anything that happens within the cell. And then the third thing that we can look at is studying the development of a certain disease. So we can look at how a certain disease develops, right? So the fourth thing that we've got now is to select traits that might be beneficial to humans. And we'll look at that, we'll go more in depth into these categories as the lessons go on, and we'll understand these. But for now, what we need to know is that we use genetically engineered organisms for these four things: to either study expression of a particular gene, or to investigate cellular processes, or to study the development of a certain disease, or to select traits that might be beneficial to humans. You can read the paragraph above to look at an example of genetic engineering, and that is where researchers inserted a gene of bioluminescent protein, and that protein is called the green fluorescent protein, or GFP, into various organisms. And GFP is a substance that is naturally found in jellyfish that live in the North Pacific Ocean, and they emit a green light when they are exposed to ultraviolet light. And the organisms that have been genetically engineered to synthesize the light, and when we say genetically engineered, basically I inserted that gene, that GFP gene, into an organism, and now they can start to synthesize, they can start to make that light. Oh, sorry, I just mixed in the lines. Organisms that have been genetically engineered to synthesize the DNA for GFP, such as the mosquito larvae shown in figure 13.3, can be easily identified in the presence of ultraviolet light. The GFP DNA is attached to exogenous DNA to verify that DNA has been inserted into the organism. And so the important things for you to know in this page are what is genetic engineering, and what are we using genetically engineered organisms for. Right, let's look at DNA tools. All right, restriction enzymes. So sometimes some types of bacteria contain powerful defenses against viruses, so they've got to defend themselves against viruses. And one way they do that is they use restriction enzymes. And what a restriction enzyme does is it recognizes and binds to specific DNA sequences. So this restriction enzyme would recognize a certain sequence of DNA, and it would cleave the DNA within that sequence. And cleave basically means it would split or sever the DNA within that sequence. Now, a restriction enzyme, also called an endonuclease, cuts the viral DNA into fragments after it enters the bacteria. And this viral DNA that we're talking about is the virus's DNA. So we're talking about how bacteria use these restriction enzymes to defend themselves. And what happens is when this DNA enters the bacteria, what happens is this restriction enzyme, also called endonuclease, it cuts that viral DNA into fragments. Now, after their discovery, so since they were discovered in the late 1960s, scientists have identified and isolated hundreds of restriction enzymes. So these enzymes come in really handy when we want to study a certain thing, right? Because we can cut that thing out. So what happened is when scientists discovered these restriction enzymes, they identified and they isolated hundreds of them so that they can use them. Now, restriction enzymes are used as powerful tools for isolating specific genes. So we can use them to isolate a specific gene, for example, gene A, gene B, whatever gene that we're looking at that we want to study, we can isolate it, or we can isolate a whole region of the genome. Now, when the restriction enzyme cleaves the DNA, it creates fragments of different sizes that are unique to every individual. And again, cleaves basically means it splits. It looks something like these pictures right here. You've got the DNA just normally double helixed, and then we split it, right? This is another way of looking at it. All right, let's look at EcoRI or R1, I'm not sure. All right, so one restriction enzyme that is used widely by scientists is known as EcoR1. I'm gonna assume EcoRI, whatever it is, we're gonna call it ECO, right? So the scientists use ECO because it specifically cuts DNA containing the sequence GAATTC. And yes, you've got to memorize it. So what does ECO specifically cut? It cuts GAATTC. You have to memorize it, okay? All right. Now, the ends of the DNA fragments that are caused by the ECO cutting them are called sticky ends. So these ends right here, they're called sticky ends. And that is because they were attached, they were complementary to something. So they contain single-stranded DNA that is complementary, because as you can see here, this DNA was just, stuck and happily living its life, and then ECO came and cut them in half, and now these parts are complementary to here. So we've got the A is complementary to the T, A complementary to T, T with A, and so on. And so they're like, but we want to be together, they're nice and sticky and ready to stick to their complementary base. And what's good about having sticky ends, or what is important about having sticky ends, is because with sticky ends, you can join them to other DNA fragments. So if I wanted to add an exogenous DNA, or if I wanted to take a DNA from another part of the organism or another organism, since I've got these sticky ends, these complementary ends, I can join other DNA fragments to these complementary sticky ends, right? Because they're sticky, I can go ahead and stick other DNA to it. So it's very important to have sticky ends. All right, an important thing to note is that not all, let me just highlight this, not all restriction enzymes create sticky ends. Some enzymes produce fragments containing blunt ends, and that is created when the restriction enzyme cuts straight through. What I mean by straight across is, if we look here, let's talk about the sticky ends again, all right, before we talk about the blunt ends. So if we look at the sticky ends, we've got strands of single-stranded DNA, right? So here in the beginning, they were all connected, double helix. Then when I split them, this part is connected just fine, they're with their pairs, but right here, they're alone, right? They're single-stranded. They've got sticky ends. They don't have a complementary pair. They're waiting for a pair because we cut them, right? We cut them, so now they don't have pairs, right? So they can pair with other things, with DNA that I've chosen, that I want to pair it with, right? So it's sticky ends because it's single-stranded, right? It's complementary, it's ready to stick to something else, it's not sticking to anything. But what happens with blunt ends is we cut right across. So we've got this piece and this piece, and as you can see, they're both double-stranded. We don't have any single-stranded DNA, we don't have any loose edges, if you will. They're all connected, right? Here, you can see they're not connected, right? This one's by itself, it ain't got no friend. But here, they're all with their friends and happy because we cut right across, right? Right across. So in this case, we've got blunt ends, and in this case, we've got sticky ends. So that was important to note. And blunt ends do not have regions of single-stranded DNA, right? So we saw here we've got single-stranded DNA because we cut it and took it away from its friend. So now it's by itself looking for someone else to come and be its friend, right? But then here, we don't have that, right? They're both with their friends, nice and happy. We didn't separate anyone. We just cut them in half. Okay, we've made blunt ends. So blunt ends do not have regions of single-stranded DNA and can join to any other DNA fragments with blunt ends. Okay? Now let's talk about gel electrophoresis. An electric current is used to separate DNA fragments according to the size of the fragments. And that process is called gel electrophoresis. So what happens in gel electrophoresis is we separate the DNA fragments according to their size. So if we look at the figure here, I'll just remove these images. Okay, if we look at the figure here, we can see how the DNA fragments are loaded on the negatively charged end, right? So we put them here first on the negatively charged end of the gel. And when an electric current is applied, so when we apply the electric current, the DNA fragments move towards the positive end of the gel. So I enter the gel into the DNA into the negative side, and once I start the current, the DNA start going towards the positive side, right? We can see them running towards the positive side, or drifting towards the positive side. The smaller fragments move faster. Obviously, if you have a something small, for example, if you take a small car toy with a huge big truck toy and you push them both with the same force, obviously the little car toy will go farther because it's smaller, right? But the truck, since it's heavier, it's not going to go that far. Same thing applies with DNA. So the smaller fragments, they move faster and farther than the larger ones. And the unique pattern created based on the size of the DNA fragments can be compared to the known DNA fragment for identification. So right here we've got something called a marker, or the known DNA. So I know this DNA, it's my marker, it's what I'm basing the rest of the DNA on. So it's what I'm comparing everyone else to. And what happens is I'll compare the unique pattern created based on the size of the DNA fragment, and I would compare it to that known DNA for identification. Also, portions of the gel containing each band can be removed for further study. And that's it for DNA tools. Okay, let's sum up the first part of the lesson. We talked about restriction enzymes and gel electrophoresis, and let's review restriction enzymes first. So our example in the book is EcoR1, and we said that its function is that it cuts the DNA strands into fractions. What is its applications? It can be used to create DNA fragments with sticky ends or blunt ends that can join to other DNA fragments. So it's very important to know that both sticky ends and blunt ends can be joined with other DNA fragments. The difference is the sticky ends have single-stranded DNA, right? DNA that isn't paired with anything because we cut it off its pair, right? It was a double helix, it was with its friend, and then we tore those two friends apart, right? And now this friend is looking for a new friend. But with blunt ends, we cut straight through. So the friends stay together, we don't cut the friends away. Now, gel electrophoresis, what it does is it separates DNA fragments by size. So we're looking at the size of the DNA. And it is used to study DNA fragments of various sizes. That is its application. And this is it for the first part of the lesson. We'll talk about recombinant DNA technology in the next part.